So we are very proud, Elias, to welcome you here in the Oncopole, and I think it's time now to listen your talk about CLL from genosis to single-cell analysis. Thank you.
So I will address this issue, this trip that we started some years ago, in particularly in chronic lymphocytic leukemia. But I think it's a trip that we are doing in all types of tumors and hematological neoplasias. We start with morphology, but we keep going with other technological aspects that allow us to better understand the diseases that we are dealing with. CLL is very well known disease with a clinical history that is very clear, very well known. And I think it's a model also to study cancer development, because it's a privilege to be able to obtain samples from the patients at different time points, and we can study, because although we know the natural history of this disease with very stable phases of the disease, the patients progress, treated, sometimes relapse. We don't know very well what are the mechanisms driving this evolution. And also from the pathology point of view, we have different morphologies that correlate with her history, but exactly what are the mechanisms that lead to these progressions are not very well known. For that reason, some years ago, approximately 10 years ago, we have the opportunity to join the International Cancer Genome Project with a large project funded by the Spanish government to study the genome of CLL. And in these 10 years, I think we have learned, and I'll try to convey these ideas to you, we have learned to characterize specific disease subtypes that have biological and clinical impact. We are starting to understand this evolution that I told you, and I think from understanding this, we are having new ideas on how to improve our diagnosis and our potential new treatments. The risk stratification of these patients is very important to plan the management of these patients according to the biology of the disease, and obviously some new strategies. I will start by this figure that summarizes the work that we did to sequencing the genome, whole genomes, whole exomes of 500 patients of CLL. And each patient is reflected in one line of this theater, and each row represents one genomic alteration, different mutations and copy number. As you can see that most of the alterations are concentrated in a group of patients, whereas there are others that do not carry virtually, or we're not able to detect virtually no alterations. But this study came out more or less at the same time that the study, also with around 500 patients from the American group in Dana-Farber and Broad Institute, and was very shocking at that time for us, because in green is the results of the number of mutations in our study, and you see in yellow was the results from this American group. And you see that we only have 36% of alterations in common, and many different. Also, an intriguing aspect in both studies was that around 50% of the patients, even we had sequenced the whole genome or whole exome of these patients, we didn't come with any potential driver. So these questions led us to think that probably the landscape of CLL was still not fully characterized, and there were some questions that need to be addressed. One aspect that we learned by comparing these studies is the heterogeneity of the disease, and important aspects like the clinical stage in which the patients were sequenced were very important. They were telling us something. For instance, you have in green were patients that were sequenced that were introduced in a clinical trial, the German CLL-8, that were patients that required treatment. They were in a clinical trial, very aggressive disease. And you see in green that were already some mutations that were really significantly higher in this group of patients that went into a clinical trial. On the contrary, our study in red is a population-based study, so where patients that were came to our hospital, and you see that some of the mutations were a very low frequency. Definitely that was telling us that the stage of the disease was determinant in the profile of genomic alterations. The other thing was MYD88. We discovered for the first time the presence of this mutation in CLL, but you see that in Dana-Farber, and it's associated with good prognosis. But we observe in our series that is related with the age of the patient. MYD88 is mainly present in young patients with CLL. And you see the Dana-Farber group that has more MYD88, the median age of this group was 10 years younger than ours. So again, is a mutation that is related to a clinical aspect of the disease. And other source of heterogeneity is, we talk about CLL, but now we know that CLL probably is composed of different, if you want, subtypes of the disease. And the major is the cell of origin, naive-like or memory-like. And this is characterized by the mutational status of the immunoglobulin. And again, in pale yellow green, you see the cells that derive from the naive-like with unmutated immunoglobulins. And you see that there are some mutations that are more frequent in this type and virtually not present or very low frequency in the other. So again, the cell of origin marks heterogeneity in the disease that will, in certain way, determine the mutations of the genome that will be selected in the evolution of the disease. And finally, just another layer of heterogeneity that we discovered in our laboratory, thanks to the work of Iñaki Martín-Subero, that is leading the group of epigenomic studies in lymphomas, was this is a methylation arrays of our 500 patients in which we compare the methylation profile of the CLL with the methylation profile of normal B cells that you have here. And we found that a group of patients on the left that had a methylation profile that was very similar to the naive-like normal B cells. On the contrary, we had another methylation profile of the CLL that was very similar to memory-like cells. But the surprise was that we found a group of cases that we have never seen before with other approach that were characterized by a methylation profiles intermediate between the two of them, and we didn't find any normal cell that had this imprint of a cell of origin. And we call this group intermediate in terms of a methylation. The relevant things, and I will come later for that, is that the evolution of these patients with this methylation profile intermediate was intermediate between the naive-like that had a very accelerated disease and the memory-like that had a very slow disease, and those were intermediate. So we were convinced that this group was a real biological subtype of disease with clinical relevance. And we were confirmed that because when we look at the different mutations in the different subtypes, naive-like, memory-like, or intermediate, it were two mutations, particularly in ATM and particularly splicing factor 3B1 that you see were significantly enriched in this subgroup of methylation intermediate. So this is the first idea that I want to convey. The genome of the CLL is probably dynamic in the evolution of the disease and is also determined probably by the cell of origin or initial aspects in the evolution of the disease. Also the very heterogeneous number of mutations. I showed these curves. You realize that there are very few mutations that are present in more than 5–10% of the patients. So most of the patients carry mutations that are relatively present in very small group of cases. When we put all that together, we see that these genes mutated are targeting some pathways that are relevant for the biology of the disease. And when we look from this perspective of the pathways that are targeted by different mutations, we see that probably this large heterogeneity might be in certain ways reduced. And the main pathways, I think, is the Notch pathway, the B-cell receptor and toll-like receptor pathway, pathways related to RNA metabolism and metabolism, DNA repair and genomic stability, So we and others, during the last years, we have been trying to clarify the different role of these mutations in these pathways. But as I told you, one of the intriguing things in our study was that about 50% of the patients we couldn't find a potential driver. So that indicates that there were things that we were missing. And I want to concentrate on two aspects that we later work more and tell us that there were more genomic alterations to discover. And one of them was in this machinery of the splicing machinery that we discovered together with the group of Lincoln-Stein in Toronto, that there were mutations in one element of the splicing machinery that never before had been recognized as a target on oncogenic target in any type of tumor, and that was the small RNA U1 that, as you know, is one of the elements required to start the scaffolding of the spliceosome to splice the different to start the mechanism of splicing. The U1 is a small RNA that binds to the splicing consensus sites and has this, and then the group of Lincoln-Stein detected that this U1 had a mutation, frequently mutated position in the 3 position that of this RNA. Why this RNA had not been discovered mutated before? Because it's a very complex gene. It has 7 genes in the genome and more than 100 pseudogenes. So the previous pipelines to analyze the genome sequence were unable to detect that. So only when they develop a new bioinformatic pipeline to identify these repetitive regions was possible to detect mutations in these areas, the complex areas of the genome. Our collaboration started because they analyzed the pan-cancer data set and found that they found these mutations in very few tumors. And particularly, you see one of the tumors that had more mutations was a CLL. And those were our cases that we have deposited in the pan-cancer repository. And so they call us to see we could confirm this data and try to see what was the clinical impact of that. And in this position, there are 2 mutations, an A2C or A2G change, and A2G was present in children medulloblastoma, and the A2C was present in hepatoblastoma. So we expanded this study with my bioinformatician and fellow Ferran Nadeu, and we compared the alterations of the U1, that is this group of patients, together with the whole information that we had from our cases. And that was interesting because these mutations, as you see, were mutually exclusive with splicing factor 3B1. So those were mechanisms targeting the same pathway, but mutually exclusive. And the other thing, they were associated with activation of NF-κB and POD1. But also interesting was that there were some patients that didn't have any other driver that we could have detected, and they carried these mutations. From a clinical point of view, it was also interesting to see that the mutations in U1, here in red, had a rapid and accelerated disease similar to cases carrying mutation in splicing factor 3B1, ATM, or NOTCH1. So it had a similar impact. Virtually all the cases carry unmutated immunoglobulins in this case. And when this is the data, as you see, of more than 500 patients, the clinical impact in a multivariate analysis was independent of other stages. So we think is a relevant mutation similar to the other ones. The problem is how to detect this mutation in the clinical practice. But we have a new information here. And the other relatively new mutations that were detected in the last years was in this B cell receptor itself, in the same immunoglobulin that is the B cell receptor that is crucial for the survival of the cell, the CLL cells and other lymphoids, the signaling through the B cell receptor. Amas Juma in Basel proposed some years ago that in CLL and other lymphoid, particularly in CLL, but probably other lymphoid neoplasias, the trigger of the signal of the B-cell receptor was not an external antigen, but it could be that the same immunoglobulins itself could cross link between them and send signals to the center of the cell. That was confirmed later on by other groups, particularly Paulo Guia in Milan, that they showed that this immunoglobulin that carry in the lambda chain was the 321 gene. They have a configuration of certain amino acids, particularly allicin and 2-aspartates, that could combine with, in the position 110, when this position had a mutation to an arginine, that created the possibility that these two immunoglobulins that were close, they could cross-link and transmit strong signals to the cells. And that was studied in a group of different cohorts of patients, and they detected between 8 and 20% of these cases carried this mutation, and those were associated with a more accelerated disease. Interestingly, from a point of the cell of origin point of view, is that half of the patients that carried this mutation had unmutated immunoglobulins, so apparently good prognosis, and half of them had unmutated immunoglobulins. But as you can see here, patients with memory-like CLL carrying this mutation had an accelerated phase. So, you know, this paper is what I call is my reclusion paper, because it was in the COVID moment in which we were not allowed, I was not allowed to go to the hospital, and I was at home, so I had more time to read, and I read all these papers, and so I said, well, come on, we have the genomes of more than 500 patients, and we didn't see this mutation. And we didn't see these mutations because the immunoglobulin gene is one of the regions of the genome that is extremely difficult to analyze with the current pipelines. So my bioinformatician Ferran there, we discussed this issue, and he had developed a pipeline to analyze specifically the immunoglobulin gene receptor by extracting the information from the whole genome and whole exome sequence. So I said, well, you have this pipeline, we have this problem, why you reanalyze our data for this mutation? And that's what we did. He was at home, I was also at home, and we were discussing that by the phone. He reanalyzed our 500 cases and found this mutation in 6% of our CLLs, but again, we could confirm that was half were memory-like CLLs and half were unmutated immunoglobulin, so naive-like. But interestingly, and I went back to one of my first slides that I said that there were 3 epigenetic subtypes. Virtually all these mutations were exclusively found in the methylation intermediate subgroup of cases, as you can see here. So we could have started to have a clue what these intermediate group cases. And these cases in yellow had a level of mutations in the B-cell receptor that was intermediate also between the other groups. And also these cases carrying this mutation in the arginine 110 were enriched significantly in mutations in splicing factor 3B1 and ATM. So this mutation in the B-cell receptor is delineating a subtype of CLLs that is very specific, I think is very particular in terms of genomic alterations. We also demonstrate other things. For instance, these cases have an activation of the Wnt pathway that is not seen in the other CLLs. So this is, I think, a biological subtype of CLL that is determined by this mutation in the lambda V321 gene. And from a clinical point of view, we confirm now in more than 500 cases what had seen before in smaller series. This here you have in blue and dark and pale blue the cases that carry this mutation that have unmutated or mutated immunoglobulin. So you can see that the mutational status of the immunoglobulin gene loses its impact when you have this R100 mutation. And also similar for the overall survival of the patients. And now when we interpret these cases of the methylation subtypes, in yellow you see the intermediate, with intermediate time to first treatment and survival. When we split the epigenetic intermediate cases in carrying the R110 or wild type for the 110 mutation, you see that this intermediate group splits, and the cases carrying the R100 really behave as the CLLs with unmutated immunoglobulins. So the intermediate clinical evolution of this epigenetic intermediate is due to a group of cases that carry more aggressive impact that are related to this R110 mutation. And that has multivariate independent analysis. So I think this mutation is important in the clinical practice because the status of the immunoglobulins is determine information for the clinicians to manage or follow the patient. So I think this is beyond the mutational status, so I think it's important. That's why we thought that we extracted this information for the whole genome sequence. So I thought we needed to develop assays to use this mutation in the clinics. And that's what, when Charlotte Sirik came to Barcelona, we talked that he could try this idea. So we have 600 new cases independent from the previous series, in which, together with Ferran Nadeu, he had the idea to develop oligospecific primers to detect this mutation in the immunoglobulin gene. It's a little complex because usually these cases carry a lot of mutations, so we have to design at least 2 primers to prevent that the hypermutations of these regions could prevent the primer to bind. But with 2 primers in the common region and 2 oligospecifics for the R100 and 1 control gene, we could detect these mutations in virtually all cases. And we confirmed that by NGS, and this assay was working very well and captured all the cases. So in this independent series of cases, we confirmed that there were half and half cases related to the mutational status of the immunoglobulins. Also, most of the cases were intermediate from a epigenetic point of view, and half of the cases have a stereotype 2, but half didn't have this stereotype. So I think this is more important than the stereotype. So then when we went to look at the clinical impact, and we confirmed that the cases having here in yellow and blue, the evolution, they have a shorter time to first treatment independently of the mutational status of the immunoglobulins, also independent of the intermediate epigenetic state, and also overall survival. So I think we have confirmed now that the detection of this mutation also is relevant for the clinical practice, and we start having assays that might detect this mutation in the clinical practice. And then, we had different results with the Dana-Farber group and ours, and one way to resolve when you have discordances is to talk and say, can we collaborate and see if we can find out where are the possible discordances. So we talked with Kathy Wu and Gary Getz, and we decided 4 years ago to join both series, 500 each, and also to increase some more cases. So we ended up, and the paper has been recently accepted, we ended up with this whole genome or exomes of more than 1,100, as you see, CLL cases that came from different studies, mainly from, in pink, from our studies, but also from Dana-Farber, also the German CLL8 group and whatever, and what we studied from whole genomes, but also transcriptome, and also the methylation arrays. And just to give you an idea of the heterogeneity and complexity of the genome of CLL. So now we have the cases of more, these 1,100 cases. We studied also the new mutations that I have described. And now we discover almost 40 new genes that we could call as potential drivers in CLL that were not discovered before. And this is because we develop new pipelines to call for drivers, but also for the statistical power. However, it's very intriguing that most of these novel mutations, or most of the mutations in CLL are present in less than 2% of the patients, what indicates the tremendous heterogeneity of this disease. And also in red were the new genes that we found mutated, but and you see that most of these mutations were found in 1 or 2% of the patients. So it's biologically interesting, but really clinically challenging to make a sense of all this very heterogeneous disease. But now we found that virtually all patients, only 3–4% of the patients, we can have genes that we can call as drivers. Also, we use different pipelines. That was mainly the work of the Dana-Farber group to call drivers based on the three-dimensional structure of the proteins, how the mutations were clustering in certain hotspots, and that's why some genes like the IS3 or MAPK27 were called as potential drivers. In any way, also, this study emphasizes how certain mutations and copy number alterations are specific of the naive-like or memory-like CLL. For instance, all these in pink are almost exclusively found in naive-like, whereas other mutations like CARD11 or MYD88 are mainly in the naive. So again, emphasizing that these two CLL subgroups are really probably two different groups of cases. And now we have expanded in red the number of genes, but again, targeting the same pathways that I mentioned before. So I think we have ended up with a really almost complete, I would say, map of mutations in CLL after all these studies. And I think the new challenge is how we start to understand the complex evolution of CLL. And to do that, we need to study the disease at different time points of the same patient. And we started with this complex evolution to that we call pathologist Richter transformation. That is a transformation of an indolent disease, a CLL, into a large B-cell lymphoma. And this is a challenge for the pathologists. And we were discussing with Pierre Camille yesterday and this morning also, because we pathologists encounter now that the patients are treated with ibrutinib, a morphological aspect under the microscope like this one, in which when the ibrutinib is withdrawn, for reasons that still we don't understand, these cells start to proliferate and become larger, like you have here. And the Ki-67 is extremely high in these patients. So I have this case thanks to a patient that provided us with this, the possibility to study this disease. And this situation, the intriguing question is that we pathologists make the diagnosis of Richter transformation in this case. The issue is that if these patients are, the ibrutinib is reintroduced, this extremely high proliferation regresses. So morphologically is transformation. Biologically, it seems that is not transformation. The question is how can we, in addition to the clinical history, can we interpret this biological problem? So I think it's important to understand well what is happening in this evolution, in this of Richter transformation. And to do that, we collected 90 patients that are here in different numbers, and in each individual that is here with a number, we collected different samples at different time points of the disease. As you see, at least 2 or up to 6 samples in a span of time that goes from, in 2 patients we diagnosed the Richter at the same moment that the CLL, but in the others, the lapsus of time was from several years to up to 19 years. And all the Richter and the CLL was the same clone. So we confirmed that this is an evolution of the same clone. And they were treated with, the first 3 were not treated because was discovered at the same time. In the others were treated with chemoimmunotherapy, and the others, in addition to this traditional treatment, were treated with novel agents, particularly BTK inhibitors. And in all these cases, we sequenced the whole genome. In most of the cases, we had also normal DNA from the same patients. And in 5 patients that we have enough samples, we also study different layers of the methylation of the epigenome, like methylation, histone marks, of activated regulatory regions, open chromatin, also the transcriptome, and also in these cases we could study single cell suspensions, and we did single cell DNA sequencing of a panel of genes and single cell transcriptome. And this is the cohort that I'm going to explain the main results. And from a mutational point of view, we detected mutations in cell cycle regulatory. I will summarize these findings because I think the integration of this data is important. Also, some of these mutations were already present from the very beginning of the disease, like a mutation in cyclin D2 that was activating. But all others, like the translocation of cyclin D3, was acquired in the moment of the CLL, of the Richter. Also, MYC was activated by different mechanisms. Notch was activated, some of the patients from the beginning of the disease, in others were acquired. NF-κB activation was also the other pathway that I think is crucial for the Richter transformation. And then a cataclysm in the genome. I mean, there were chromotrypsis, chromoplexia, a lot of new translocations that were acquired. So it's a major alteration in the genome. If we summarize all this, I think some interesting ideas come. If you put together all the cell cycle alterations, you see that virtually all cases have oncogenic mechanism to activate the cell cycle. Second, if we look at MYC, virtually all the patients have a MYC alteration, and the ones that do not have MYC oncogenic event, they have Notch activation. And you know that MYC is a downstream of the Notch pathway. So, as you see, it seems that all Richters require a cell cycle oncogenic alteration plus a MYC alteration. And also you know that MYC conveys proliferation and growth pattern, but also is a strong signal for apoptosis. So these cells might require oncogenic events to give strong signals of survival. And probably that's why they are selecting NF-κB activation mutation. So I think this summary gives us an integrating idea of what might be happening in the transformation of Richter. When we look at the different layers of alterations that I show you, mutations, copy number alterations, structural variants, DNA methylation, opening of new regulatory regions, or look by the histone mark or the ataxic, you see that there is a progressive complexity between the original CLL, the CLL samples that relapse after treatment, and the final Richter transformation. If we analyze these different layers, we are learning different things. Copy number alteration and structural variants. I told you, this is a circus indicating the translocations between the different regions of the genome. And you see this is not the CLL karyotype. I mean, this is a tremendous alterations. And particularly this phenomenon of chromotrypsis that are targeting different shuttling cutting different chromosomes, are targeting important genes that are relevant for the biology of the disease, like the ones I show you here. And keep this idea, because there will be some interesting information that we will show you later. And I think that that observation made me go back to what I told you, like pseudo-Richter. This is one of the two patients that we have seen, and this is the CLL. This is the tonsil in which one pathologist diagnosed as Richter transformation, and probably I will have said the same. You see it's the same clone. But when we look at by arrays to the karyotype of this case, you see that there is not a single gain in this proliferation in the tonsil that we pathologists call Richter compared to the original CLL. And this is totally contrary to what I showed you before. And in the other patient that I saw, this is a CLL, and this is the lymph node that was also diagnosed by me as a Richter transformation. There is not a single gain, and there is not a single gain in the mutational profile. So here is something that will be incompatible with what I said before of the Richter, of the tremendous complexity that is acquired. So I am starting to think that these studies could help us pathologists to distinguish these proliferations that are morphologically like Richter, but they are not Richter. So if we analyze another layer of complexity, is the profile of mutations. You have in pink the Richter syndrome cases, the Richter samples, and in gray you have the CLLs, including the original CLLs, but those are our series. And you see that the mutational profile of Richter is different from the CLLs completely. So our bioinformatician Ferran and Romina look specifically at the signatures that they could extract from the mutational profile of these genomes. It was very interesting because they found some known mutations, but they came with two mutations that had not been previously described, two mutational signatures. And one was this one, in which I don't know if you are familiar with these diagrams, but this peak represents a nucleotide change, a C4A, that is in the context of a previous C and a subsequent A. So this mutation of a C4A in this context, we didn't know what was that. And they told me, we found an artifact that we don't know. But whereas the paper was in review, a paper came out describing a mutational signature that is introduced in the genome by ganciclovir. So the patients that are treated with ganciclovir, this agent introduce multiple mutations in the genome that has this particular change. So we went to look at the history of this patient, and indeed this patient had received ganciclovir. So you see how treatments might leave an imprint in our genomes of the diseases. But that has another consequence that will come later. But also there was these very peculiar mutations that were T4A or C4G changes in the context in a subsequent A that we didn't know what that was. Has not been described. So as we see that this mutation was related to a therapeutic agent, we thought could be that this patient in Richter had some particular treatment that has introducing these mutations. These mutations are here in red, and what you see is they are acquired during the evolution of the disease, because in any of the patients we found these mutations at the beginning, and was at the Richter sample, but in some patients was also in samples before we could call Richter. So we plot all the treatments of these patients, here in all the different patients and the different treatments that they have received. And you see the patient that have received the ganciclovir had this mutation. But then we have 3 patients that have a signature that had previously described as introduced by melphalan. And we found 3 patients. 2 of them had received melphalan. So again, an imprint of the treatment. And intriguingly, all the patients that have this new signature that we have described, they have received either chlorambucil or bendamustine. So we think, and we did other experiments to show that probably these mutations are introduced by these agents. Again, an imprint of the treatments in our genome. But this is interesting, but that led us to another idea that were conflicting with other ideas. If you think that these agents are acting in our genome in a random way, and if you think that to detect a mutation we need several reads, if we are detecting these mutations that have been introduced randomly, that means that the sample in which we have detecting this mutation is coming from the expansion of a cell that in the genome carries the same mutational profile, because otherwise we will not be able to detect it. So that made us think that the clone that gave rise to this Richter, if you saw this mutational profile earlier, that probably meant that this clone was earlier already being developed and expanding in the CLL. And also by informatic analysis, the bioinformaticians could group different mutations, and they can elucidate the clonal architecture of the population based on association, a statistical association of different mutations. And they can build these phylogenetic trees, in which here in this particular patient, you see that they detected two clones late in the evolution that have this signature. So it seems that this signature associated with Richter were developing in two different clones. And they built this fish plot that I think are fascinating. Each color represents a subclone, and each time point, 1, 2, 3, 4, 5, are different time points of the disease from time 0 to developing of Richter 19 years later. And we have these different samples. And you see that the clone that is present in the Richter, they predict that probably was already at the moment of diagnosis. And you see that different time points, there are different clones, but all of them go to the beginning. They predict that already they can detect this association of mutations at the moment of diagnosis and different mutations. But in this particular patient, we have flow cytometry of different time points. And in time 4, you detected, that was here, that we have not detected the Richter morphologically. You see that by flow, there is in green the CLL's clone, but in pink, there is a group of cells that are larger. And they have a different phenotypic profile. On time 5, these cells have regressed a little bit, but another group of cells come here, and these cells are the ones that are being expanded at the end. So this idea that we could reconstruct based on the informatic analysis of the genomes, we could confirm by flow cytometry that at least at time point 4 or 5, we could detect these subpopulations. And by the scatter site, these cells look like larger. So probably there were like two clones that were developing at the same time. So if we put all the cases, what I want to summarize here, those are the different time points in peak, is that in at least 5 patients, we could detect this, or we could predict or infer, that will be the word, that there were already clones carrying the Richter signatures early before, or the Richter mutations early before the diagnosis, and in 5 of them at diagnosis. This idea was very intriguing, very puzzling, I would say, and so we decided that we need to confirm this idea in a more solid way. So we switched to single cell, and the fellow that is doing that, he likes to give me these pictures to show what bulk analysis means versus single cell analysis. And I like better this one, in which I think by analyzing single cells and putting them together, we can capture the heterogeneity of the reality that we are seeing in a better way. So we had samples from some patients at different time points, and you see that by informatic analysis we could detect some mutations that are present in the trunk, but others, like now I want to concentrate on the P53, in these two mutations of P53, in this 245 codon and this 195, that is present in the pink clone here, and the one that is expanded in the Richter, the 245. So you use single cell analysis of these different time points. And what we detected is that both mutations that are in different cells, in different clones, were already present 14 years before they expanded at the moment of diagnosis at 0.1% of the cells. So these cells with these mutations that expanded at time 4 and time 6, they were already detectable at the moment of diagnosis. Then we did a single-cell transcriptome analysis, and this is the CLL samples of case 12. And we detected different clones that I will not go into details, but I think we represent the recirculation between the cell and the lymph node. And then the transcriptome of the research cells was clearly different. So when we went back in the separate samples, you see that the transcriptome of the Richter was detected with very few cells, also at the moment of diagnosis and at different time points. So these cells with this transcriptome were especially detectable at different proportion, different time points. So we have transcriptome that is of the Richter is detectable. Some mutations were detectable. The question was, could be that also this cataclysmic of chromotripsis could be also detectable in these cells? And to answer that, we use the power of the transcriptome to interrogate how the status of the genome that is coding for these genes is. So by integrating the expression of the genes that are in a particular region of the chromosome, you could detect if they are simultaneously amplified or simply overexpressed or totally downregulated. That will indicate that this chromosome region is lost. And this is what we could detect. And look at this patient, this is the Richter. These regions that are present in a chromotriptic region that we have detected by bulk analysis were also detected in cells at different time points of the disease, including some cells at the very early evolution of the disease, diagnosis and early time. So that indicates that already at the moment of diagnosis, these cells not only carry the transcriptome, not only carry the single mutations, also could carry these chromotriptic alterations and also some else. So the final layer was the epigenome. We look at the methylation. There are no major methylation changes between CLL and methylation. There are some, and Richter, but there are changes in the open chromatin regions and the transcriptome. So we look in details that this is the activated regulatory regions in Richter here. We discover several regions that are totally different, open chromatin activating enhancer promoters compared to normal cells and CLL. When we look at the regions that are contain these new open regions, we detected that they have consensus for the binding of certain transcription factors. And the first one was TAD4, MAS, A2F1 related to proliferation. And that's interesting because TAD4, we didn't know, we learned that it seems that a transcription factor that is important in the regulation of mTOR and certain metabolic pathways. When we look at the differential expressed genes, that makes sense because the genes that were highly regulated were related to proliferation with MYC and the pathway. When we integrated this, we saw that the genes were in pathways that were controlled by these transcription factors. And among them, I want to emphasize the high expression of genes related to OXPHOS pathway, phosphorylation oxidative, and downregulation of the B-cell receptor pathway. And we confirmed that by gene set enrichment analysis. That was in Richter, but what happened with these particular pathways in the cells at different time points of the disease. And again, you see that the OXPHOY, in these cells that we have defined as Richter in the early times of the disease, also they were enriched in the OXPHOS, and they were also downregulated the B-cell receptor pathway. So this view of high OXPHOS and low B-cell receptor, they were already in the cells that we have defined as Richter cells 19 years before the diagnosis. So is this just a bioinformatic finding, or is that real? And that's what the reviewers asked us, and it was good because gave us an idea that here you have in pink the Richters, and you have here the CLL cells from the same patient. And you see that the activity of the OXPHOS is significantly higher in the Richter compared to the respective CLL of the patients. The exception, well, in this was less manifest, although, well, I will not go in detail. But this patient has a mutation in the R110 that I explained before. So, and then the B-cell receptor is only a expression problem, or is really functionally the activation of the B-cell receptor is diminished. And you can see here in pink the Richter. This is the calcium flux after stimulation with IgM. And you see that the Richter cells have a decrease in the B-cell receptor activation when we stimulate with IgM, except the case that has the mutation, the R110, because this mutation induces the signaling of the B-cell receptor independent of any external stimulation. Could that be interesting for therapy? Well, we inhibit the phosphorylation oxidative pathway, and you can see how the proliferation of the Richter cells came down after inhibiting the phosphorylation oxidative pathway, indicating that this could be a potential target for these patients. And also, we took advantage of this series of cases that were published by the Kathibu group early last year, in which they have one case of Richter, that this is their case. And we look at the OXPHOS pathway in this case, and we look at the B-cell receptor compared to the CLL of this patient, and you can see that, again, in this patient, the OXPHOS was high, the B-cell was slightly lower. And also we look if in this patient we could detect the transcriptome of the Richter, and we did. So in a case that is from another group. So I think this information is confirming our observation. So in conclusion, I'll try to convey to you the complexity of the genomic alterations in CLL, how these mutations converge in certain pathways, how the mutations in the non-coding region or in coding regions that are complex to analyze, like the immunoglobulin genes, are important biologically and clinically. The tremendous complexity of the Richter transform that, again, converge these alterations in certain pathways. And this idea that the evolution of CLL, and particularly Richter, could be already present in cells very early in the evolution of the disease. We call them the seeds of the evolution of the CLL, could be already present at very early moments of the disease. And I think we have identified pathways like OXPHOS that could be interesting as a potential targets to consider therapy in these patients. All this, you see, has been a tremendous work for many people from different institutions. I would like to thank particularly Ferran Nadeu, is my young bioinformatician, extremely talented, together with Romina Arroyo and other people, and the collaboration of Dolores Colomer with the functional studies, and Iñaki in the epigenomic analysis. Thank you very much for your attention.